My name's Boris Hinz, and I'm a professor at the University of Toronto, and I'm a Keenan chair in fibrosis research at St. Mike's Hospital in Toronto. Do I have hobbies? I do collect old records and old CDs, which for the young people, these are hard copies of MP3 files or MP4 files. So, yeah. I guess that's my hobby, and that aligns with how my brain works. So I like to put them in orders and sort them by all kinds of means. Drive my family crazy with that. My favorite molecule? Oh, that's a weird one. I'd say if you extend it to a protein, I'd say it's smooth muscle actin. It has to be. So it's yeah. That I developed. I guess the one thing which I think conceptually we did in the past is we showed, that's almost 15 years ago now, that a growth factor TGF-beta can be activated by mechanical forces. So it comes with a latent complex, and cells can use integrins and pull on the complex and release the active growth factor. There's other stuff, but I think conceptually this is probably one that I'm hopefully going to be remembered for. As you will see in a talk tomorrow I'm going to give, it's going to be plastic dishes with a rubber coating, with silicone soft dishes, if you like. That's the most widely used. What brought me to next? So I trained back in Germany. I was in a dermatology lab, and I was always interested in cytoskeleton, so how the cell is organizing itself. And like a lot of what the cell is organizing is dictated by the matrix, so that naturally brought me to matrix biology. Well, I guess we all have, so I obviously had mentors during my PhD time. They were helpful, cool guys, but I think the one who actually just gave me that independence or independent thinking was my postdoc mentor, Giulio Gabbiani. So I was at the University of Geneva, and he was the godfather of myofibroblasts, contractile fibroblasts. And when I joined his lab, he was not close to retirement. Actually, he was close to retirement. Who am I kidding? So that enabled me to take some of that, what his lab was doing hardcore, and build my own lab with it without any overlaps. Matrix teachers so because I do mechanobiology, I sort of say that way. I don't necessarily think matrix molecularly in the sense of which matrix molecules. I do think about them, but that's not my approach. So people who influence my thinking in mechanobiology is Yuli Wang, who kind of invented the idea of putting cells on differently stiff substrates, including that Dr. Harris, who came up with an assay called the wrinkling assay. I think he must have been the first to put cells on silicon elastomers, and then people follow those paths. Paul John May, so biophysicists and chemical engineers. Dennis Disher, I would name, so big names in the field of mechanobiology. Again, being matrix, being an elastomer, not necessarily collagen or fibronectin. Yeah, that's a tough one. Well, it's either a one-hour lecture or a simple stupid advice, which is that don't stress out. That'd be my main answer. Just don't. You see a lot of people very stressed for good reasons, but it's usually not helpful. So don't stress too much. Just take everything a bit lightly as much as you can, given the circumstances. Oh my! So the problem here is I have a really bad memory, but I do remember having been at ASMB meetings, not very frequently, regularly, but not frequently. I think so. There are collaborations from previous ASMB meetings, including one with Tom Barker, for instance, who's involved in the organization of this one here. So I can't pinpoint the single meeting where that would have originated. I think it's more kind of a priming phase where just repeated injuries led to collaborations between us. Cats or dogs? Neither. Tell you why. We have a cat, and turns out I'm slightly allergic to cats, so I don't touch it. I do like cats for their independence, and dogs just shit into our front yards, and I don't like them for that.
Okay, no. Fair enough.
Cheers.